Baker Ruskinn as the Director of Scientific Applications. And even though I'm in Finland, of course, the role is very global. So hence being here today. And we do want to work with the global community, which you guys are very much a part of. So we're going to talk about exosomes, and we're going to talk about oxygen biology, and we're going to talk about stem cells, and then bring all of that together. I don't kind of like expect that I'm going to educate you guys, because I think you guys know a lot of these things, but rather, like Andrew said, like inspire. Because we are a scientific community, and we want to work with you. We work together in making things, available and better and precise. So don't expect me to you guys are way smarter than I am, and I'm just going to be kind of like, let's have this discussion and go away inspired and understanding that what we do really does matter. Now, we decided to talk about exosomes today, and this is interesting because I usually talk about hypoxia and oxygen and stem cells, yes, but exosomes is a really cutting-edge thing, and I was kind of like surprised for the topic because it's not something. We just came back from a big hypoxia meeting in Ireland 2 weeks ago. And it wasn't really a topic there. It wasn't the exosomes aren't. But when you think about what they are and what they do and the potential that they have, it's just the next step from stem cells. So that's what we're going to kind of just touch upon today. What I would like to the structure here. So I'm just going to briefly, this, what exosomes are, what they do, what the potential is, and also where we get exosomes, and that kind of starts to relate with the company Baker Ruskinn, because what we focus on is providing the atmosphere for cells, and especially focusing, other things too, but oxygen. And I'm going to explain, recap for all of you why the oxygen is so important and how it relates to exosomes and how it relates to stem cells. There are a few problems, we're going to touch upon those problems. Then the problems do relate to MSCs or stem cells in general, and then we're going to solve the problems. Also a little bit of talk of hypoxia, because obviously that's the field that I come from. I did my PhD in Finland in the medical facility of my university, under the guidance of that said dude from UK, who then later got the Nobel Prize and all. So that was pretty good. It was a good career move for me too, because it's good to have a Nobel Prize winner as a mentor. Yeah, I'm going to show his photo later in the talk because I just have to do it otherwise. So, on the contrary, what usually these talks are, like just go through slides and just follow the slides. I'm going to give you a little bit of a synopsis what you're going to, not convinced, because I know you guys are believers. You're knowers. You know this stuff, but just rather, So, with exosomes, exosomes are full of potential. Now, they are things that come out of cells, right? And they are secretome parts, right? Okay, and they contain things. They contain nucleic acids, proteins, transcription factors, anything that a cell contains. But through a budding or vesicle process, the cells push exosomes out. For the longest time, I'd say 20 years, we thought that exosomes are cargo just for trash. And the cells just push trash away. But now we know that's not the case. We actually now know, like all parts of the secretome, exosomes specifically have great potential for treating conditions. Also, the potential for used as markers of disease conditions is huge, of course, because you can read what they contain. Why they are so specific? Well, because they are very specific to the cell type they come from, where they originate. So they are kind of the signature is the cell type where they come from. So that's one fact. So then exosomes come from cells. This team here is very experienced with stem cells. Mesenchymal stem cells are probably, I'd guess, the ones that are used most in research, and we understand probably most their function. So all cell types secrete exosomes. We can get exosomes from all bodily fluids, just easy. But then when you think about the potential that we know and use mesenchymal stem cells, for instance, now we know that they can be used as treatment, okay? So therefore, if exosomes are like the cells where they come from, then the exosomes from MSCs are like exosomes. Good? But exosomes have, MSCs, stem cells have problems. It's pretty hard sometimes to get enough for treatment. They are pretty immunogenic. They do often cause problems in the recipient. It is quite hard actually to get high concentration of MSCs into the target tissue. So all these things are the problems. Now going back to exosomes, exosomes don't have these problems. So they can be quite easily administered in quite concentrated form into the tissue where we want them to. They magically have very little of immunogenicity. They don't cause problems in the recipient. And they are, that's what the science tells us, they are super potent. So if the MSCs are potent, exosomes are as potent in treatment as the MSCs. Now, with exosomes, of course, it's not a perfect solution. The biggest problem with exosomes to date is that we know next to nothing of them. That's one problem. The second problem is that also their yield, when we try to collect them and multiply them, it tends to be quite low. Now, even though we know quite little, we do know through stem cells, we know one of the biggest factors of getting good quality MSCs, good quality exosomes, is oxygen. So the oxygen level we now know, it controls the quality of exosomes. So therefore, the quality of MSCs and exosomes both are affected by the levels of oxygen. Why this is that, well, as you know, oxygen is highly reactive. Explosion, combustion engine, all those things, it's a highly reactive molecule. And in cells, oxygen sensing works through enzymatic responses and changes. Now, enzymes are quite fine-tuned to work in a very narrow range of oxygen levels. So when we breathe air here at the sea level, that would be about 21% of oxygen. When we put cells in an incubator, that's 19.8. All of this is massive, like it's like fueling the burn, the burn that happens in cells, which is too fast. Now, like if you burn a fire, well, you don't have to do that here in Malaysia, clearly, but in Finland you do because it's pretty cold. If you just put too much wood in the fire, it's going to burn, but it's going to not be clean burning, right? If the wood is not clean, if it's moist, it's not clean burning. You create a lot of stuff that you don't want to create, right? Same thing with cells. If you fuel them with too much of oxygen, they're just going to burn, but there's also going to be a lot of damaging molecules, which are redox and other molecules. And they are highly, highly damaging to all biomolecules, right? So, taking that together, MSCs, by default, they come from conditions in the body where there is very little oxygen. Okay, so a human body does not contain 21% of oxygen. You know this if you stand in the wind and the wind doesn't go through you. We have very good barriers, we have tissue and everything. Oxygen does not travel very far through tissues or in liquid in the body. So in the zones where we get MSCs or other stem cells, any cells, the oxygen concentration is maybe tops 11% in the lung epithelium, and when we go down to bone marrow, it's close to 0. So hence, to get good quality MSCs, we need to keep them at all times, and I'll show you why later, at all times in low oxygen. If you want to get good quality exosomes that work like MSCs, but easier to handle and better, we also have to keep them in low oxygen at all times. So, let's take that again. The whole thing is the same. So now you know what I'm going to tell you, so now you can just follow through. Easy, yes. So repetition. So exosomes, they're small. I mean, you have endosomes and you have exosomes and you have all sorts of things, but they are quite small. Okay, so about 100 nanometers. We know that. Like I said, they are part of the secretome that comes out of the cells. They just push the stuff out. Endosomes, that's the origin of exosomes, also other vesicles as well. Like I said, they vary. They're very particular in the content of what's in there. So I told you that what's in an exosome, what the readout, if you study exosome, it depends on the tissue of origin. That would be too simple. Of course, it depends on the peroxide signaling with other entities in the cell and other cells and other exosomes. So highly complex. And like I said, this is a field that hasn't been studied. And this is why I like you guys, because you guys are actually in the very front line. I was just, coming from Europe, and I didn't hear anybody talk about these. So it's like curious. We were at a cancer meeting, and we didn't quite, next to nobody talked about these. So that's fantastic. So it contains everything that the cells contain: nucleic acids, proteins, lipids, metabolites, everything specific to the cell type where they came from. You can use them as a marker for disease. We can do that already. Lots used in liver conditions, for instance, but also they can be used as tools for taking small molecules into cells, because they also are very prone to going to a certain specific target tissue. So therapeutic possibilities. So what we do know, they too, you guys probably remember, like 20 years ago, Weinberg and others published the groundbreaking kind of map of hallmarks of cancer. Now that has evolved quite a bit, and we also have hallmarks of exosomes. So these are all the things that exosomes have been now reported and shown to work in. So when you look at the list, it's hard actually to find anything that doesn't, anything actually in the cellular life and cells function that they wouldn't affect, which makes sense because all cells produce them all the time, different quantities. They're secreted into all bodily fluids everywhere. All things living actually secrete exosomes, so that's pretty neat. Like I said, they are very specific when they find their target tissue for therapeutic purposes. It's quite easy through electroporation, to introduce stuff into the exosomes, and it's a lipid bilayer, so it's very easily going in. Again, like I said, very easy to increase the local concentration of the drug using exosomes. The side effects thus far have been very minimal. Obviously, it's a cell-free system, so the body doesn't react the way it would otherwise. It's very is a big word. Quite easy if you are capable of engineering them. So of course that's good for production type of usage of exosomes. So good. So now we have established exosomes is a good thing. We know where they come from or what they do and what they contain, and we have to get them from somewhere. And so you can get them, like I said a few times already, you can get them from anywhere. So if you have access to anything, tears, saliva, you can get exosomes. Stem cells, equally, you can get them from various places, as you guys would so very well know. I don't have to list these for you. But I have to highlight again here that depending on where you have gotten the exosomes, if they are from stem cells, for instance, the exosomes reflect that origin. Okay, so you can't just mix and match. You have to be very well aware where they came from and characterize them accordingly. Also, very important here when we go into talking about using them and the production and applications is to understand that the conditions of the source is important. So, and this, of course, I mean that when we collect stem cells where we get exosomes from, if the stem cells, when they come out of the source, are treated in a wrong way, then we cannot very well expect to have any good quality exosomes that would function as the stem cells. You know, it's kind of like, yeah, it makes sense. It makes sense. So the conditions, and as we do now know that oxygen is one of the biggest factors that goes wrong when we use these types of materials, well, then we are much further ahead of anybody out there doing the work and trying to solve these problems. So, as everybody knows, MSCs are quite capable of becoming anything, okay? They only do so, by the way, if they can maintain their stemness, their pluripotency, easy to lose, by the way, by growing them at too high oxygen. That's very well described. So, on the contrary of not knowing a whole lot of the exosomes, the MSC-derived exosomes specifically seem to be quite good in tissue repair, and this will be important for Regen Med audience here. They also have been shown to suppress the immune response reactions, massively good when we talk about these sorts of treatments. This is what we want to see. And so cell-based therapies, transplantation techniques that we traditionally use can totally be avoided by using these. So, taking things a little bit together, taking exosomes from MSCs that you guys work with. So that is a field that is probably the best known in the field because a lot of the stem cell therapy focuses on MSCs. You know that what they are can be, yes, they can be isolated from wherever and differentiate, but that, very importantly, they do the MSCs, like I said, they secrete the exosomes, which contains cytokines, chemokines, and the growth factors. So this is what we want to do. Now, how we do it, we're kind of moving towards the how and the application side of it. So you know this, you have the mesenchymal stem cells. This is, by the way, this is how the stuff is usually done, right? You know the procedure. This is the same procedure. You have them, you isolate expansion, you collect exosomes. How many of you actually work with exosomes, by the way? 1, 2, 3, 4, few. Okay, and everybody else is going to. So this is the traditional way. You centrifuge a little bit, you know, just microfluidics and filtrations, and you use them for then treatment. This is the how to do it traditionally. However, I'm kind of here to say, well, don't. This isn't really good because what I just said in the intro and mentioned a couple of times, the oxygen thing is really super important, and I know this audience knows this very well. But I'm here to propose something different because of the problems that I mentioned. There are problems. So the exosomes, yeah. So poor yield. That's the problem. You don't get enough of them. So that's. There has been papers I just checked. There are small molecule modulators like methyl dopamine and norepinephrine that you can use to increase the yield, but it hasn't really solved the problem of the yield or low crafting potential. So kind of a shame that we have this tool that we know is pretty good, but then at some point so the biology of the data says that yes, you go ahead and use exosomes, but then when you go and use them, they just doesn't quite cut it. It doesn't quite work. And so kind of a shame. Also, what literature now shows is that the major enhancer of exosome secretion is the level of oxygen. Okay, so it needs to be low. So if we keep these tools now in the incubator at 37, at 19.8, you're not going to have a whole lot of exosomes or anything for that matter. So cells you do have, they do grow in high oxygen, same as the burning kind of metaphor that I provided for you. And the burning does happen, but it's not really good, and it creates side products that are not good for you to breathe. So how and why? What has oxygen to do with the secretion? How does that work? So not a whole lot is known about this, but some work, and I have to say I've been very active in checking PubMed for the latest publications. But there are quite, I just chose stem cells here, and I chose this one set of data because this is what you constantly see in the literature when you go through how does oxygen affect. So clear, white columns are 21, and the black ones are 5% of oxygen. So, and you can clearly see that in all of these used stem cell types, growing the cells in low oxygen increases the proliferation of the MSCs. So this is where we start the MSCs, because we want to get the exosomes from the MSCs. And you can also see, to a trained eye, you can see that they are much nicer looking, and as they should be looking if you are very working a lot with these types of cell lines. You quite quickly your eye becomes trained, and you know how they should look like. So, like I said, stem cells reside in low oxygen, and so do exosomes. So they are stem cell niches, you know this. Different niches exist. The neural, the hematopoietic, the mesenchymal, all of them have this thing in common. So all of them have very low oxygen, and it goes down to 1%. So forget about the incubator stuff. This is how to do it. And why? Now I'm introducing a new thing. It's called HIF1, so hypoxia inducible factor. So why I introduced this is that molecule, that one transcription factor, amazingly, is the one governing factor of all the transcription. The other ones do a whole lot too, but HIF is the major transcription factor that affects or dictates how a cell responds to oxygen. Okay? And here, just showing events in stem cell life. Some are bad, oxidative stress, metabolism. Of course, we want stem cells to keep their self-renewal and proliferate capacities, stemness we want to keep. And HIF is, through certain enzymes and proteins, the key factor that governs all these events in cells. So, taking from this slide, HIF is something that we need. Good. Just a little bit of a sidestep for stem cells. We were talking yesterday actually about checking stem cells and whether HIF then would be the marker to look at. And interestingly, here I say no, don't look at HIF, but look at the downstream proteins of HIF. Now, HIF activates about the whole genome. Okay, amazingly, always activation, hardly ever inactivation or downregulation. We don't know why. But look at when you have your stem cells, which you get the exosomes from. These are the markers. For instance, they're stem cell markers. You know these. You know them very well. And so this data, set of data and loads others, show that if you have these, that's 24-hour hypoxia, 24, 24, 24, all of them checked for how they grow. And interestingly, you can see for here, it's for SOX2, which is the major stem cell marker. Clearly, if you see activation of SOX2, you know that your stem cells are still, you can use them basically. So you can see at 12 hours, quite a peak. Same with OCT4, the peak appears at 9 hours. Here we're talking about 9 hours again for KLF4. NANOG, very important as well, comes up at 9 hours. Same here, LIN and CD133. So as you can see clearly throughout the panel of stem cell markers, low oxygen is where the stem cells keep their pluripotency. Hence your exosome production would be high in these cells. And also the same there. Looking at exosomes then, specifically, if one is not convinced from the stem cell, this is a kind of a take-home from all the articles out there, how the secretion of exosomes occurs. So in blue, we can see that's the normoxic 21, and then in red we have the 1% of oxygen. Now these are PCA cells, but the same is true for any other cell type. And you can see super duper significant increase, so they must have quite a set of data. The secretion of exosomes in low oxygen is clearly increased. So, and this is obviously because the cells of origin are doing so much better. So, just shifting gear a little bit, and we're talking about then the low oxygen. So that's clearly the whole thing that's in the heart of this talk. And like I said, this is where my academic studies have been conducted. And why these are rather simple slides, and some of you have seen them before, but it never hurts to recap a little bit. So when we talk about the terminology, we talk about hypoxia, we talk about normoxia, and this is what you see in the literature, which is kind of like, really shouldn't be that, because we should be actually talking about, physoxia, or physiological oxygen. Okay? Physiological oxygen. That means the only range of oxygen levels in cell culture, in experimentation, that produces relevant data, where the cells live as they would in the body. Now, it's different, obviously. Normoxia that we often use, myself included, goodness, I worked for 15 years in growing cells in a CO2 incubator before kind of like the AHA. That refers to the atmospheric content of oxygen, which is 21 or wherever you are. If you're up in the Andes, well, then it's different, but 21. And hypoxia is related to malignant conditions, like in cancer, where the vasculature is just not working, or shunts, and there's a lot of reox and stuff. So hypoxia is a 1% or less. Okay, so clear definition there. So what we kind of want to emphasize, that the physiologically relevant condition is the place where your stem cells like to be, and they will function as stem cells should function in order to be manageable. And if not, then that's just throwing money in wherever, because they won't be able to perform. So just a quick check. What is the level of oxygen in the body? I'm talking about tissue oxygenation, not blood oxygenation. That's a different set of things. Somebody once told me, because I talk about 21% of oxygen, and somebody told me, hey, miss, sorry, if a person has 21% of oxygen in their tissues, they're dead, because you have to have like oxygen saturation of 98 or 100 or something. But now that's different. That's oxygen saturation in the blood, where hemoglobin is carrying the oxygen molecules, where the 4 oxygen molecules, is 100%, and that's saturation. True, if you have 21% of oxygen there, you're dead, clearly, so that doesn't work. But this is a different thing. This is tissue oxygenation. So, the air that we breathe, 21% of oxygen, very good. We need it, otherwise it's enough, because it needs to be pushed into cells. But as soon as it hits the lung alveoli here, it lowers because the oxygen diffusion distance is extremely short. It's passive, it doesn't want to go into places. And this is because you can't, because it's such a potent destroyer of biomolecules. So if we would actually have 21% of oxygen in the cells, we would, well, it doesn't work that way. That's not how the kinetics work. So we have like arterial blood, that's the first phase or place where oxygen enters, but then when we go to brain, lung, liver, clearly quite low. And now these are the places where you might get the stem cells from. Talking about organs then specifically, you can see the range from lungs, somewhere obviously hard to say like a precise number. If you want to go and do the experiments, you will have to test. But it goes all the way in retina, it's close to 0, and in the bone marrow. So this is where cells come from, and this is where we have to keep them when we work with them. It's not easy. It's not easy. It would be so much easier to just do what we always do in a BSc, and you pour a little bit here and there and pipette a little bit here and there. But then we might as well not do it, because the work, while we do get data, which is probably, it may not be wrong, but it is compromised. The data is compromised. And all scientific work relies on the fact that somebody else somewhere else can independently reproduce your work. And so doing the cell culture work, for instance, in wrong conditions, destroys that scientific, that's one of the hallmarks of scientific work that we have to respect in order to produce good quality and translatable results.